have to have my supplies, and so some of the supplies that would be required at this point in time would be CO2 that comes in through the stomata. And of course you also have NADPH that serves as the reducing power. So between your CO2, your reducing power, which that's a fancy way of saying your H's if you think about it, that's what happens in reduction reactions in biology, and energy, you could start making, with those three items, C6H12O6, or some other type of sugar, right? You could start building because of your energy, adding your H's, there they are. With the CO2 that's come through your stomata, you could start building things up. What is this the direct opposite of? What? Specifically, what part is the Calvin cycle the direct opposite of in respiration? Glycolysis. TCA, citric acid cycle, also known as the Krebs cycle. Because remember, in the Krebs cycle, you're blowing off your CO2s, and now you'll see here you're incorporating them. What's next? Some good questions about the differences between prokaryotes and eukaryotes. What type are you? Okay, good. That means you don't have a nucleus, right? That's false. Good. So a prokaryote would be lacking a nucleus, so no nucleus for the prokaryote. But of course that would be yes nucleus. And what I like to do is I usually like to go through prokaryotes and just kind of mention like all the different items that prokaryotes have, and then say that basically if you don't see it in this list, then they don't have it. So ribosomes, they have a membrane. Remember, the membrane, of course, is made of phospholipids. They also have a wall. What's their wall membrane? What's their wall made of? A lot of times, something called peptidoglycan. Y'all remember talking about that? Peptidoglycan makes the cell walls outside of bacteria. It's good stuff. That's basically all they have. They certainly have no membrane bound organelles. But certainly the eukaryotes would. They do have, of course, everything you see over there. They have all this and more. Correct? Now you'll notice that the components that they have are like the most essential items, and I want us to focus on the essentials that you would find between all living creatures, and it's kind of the topic that we've talked about more recently in the class, and that is this idea that everything has DNA, which would be genetic material. They have an ability to make mRNA, and then of course what do they make from the mRNA? Protein. Where does that mRNA to protein reading situation come into play? What structure? Ribosomes. So you see that that's something that's in everything, and that's something that's in everything, right? It's like basically taking a prokaryote would be like having an old TV, and a eukaryote would be like having a new TV. I mean, there's a power button, and there's the channel changer, and the volume control, and that's about it. Would you still have those items in the new TVs? Yeah, because you obviously need to be able to change the channel and to turn the thing on and off, correct? But you see a lot more. There's more specialization. So things seemingly become more advanced. But that component of like being able to receive TV signal and then interpret it, that's an essential item. So just like those are essential items. Cool? Good? Yeah. Let's talk a little bit about some biochemical reactions. We talked about this in pretty lengthy detail, I think, back in the day. But let's talk about two types. Let's talk about catabolic reactions. And what was the opposing one? Anabolic. If you have a problem remembering that, just think of anabolic steroids. Someone that takes anabolic steroids, do they get bigger or smaller? Bigger. Okay, so anabolic would mean to build up. I'm not saying that I'm not saying steroids are good. I'm just saying that that's a memorization technique. FYI. Catabolic. This means to break down. Anabolic. This means to build up. In which case would this require energy? Anabolic or catabolic? So it requires energy. Which one would therefore release energy? Okay, good. So when you break down food, that's going to release energy. Okay, cool. In which case would it be dehydration? Very good. And then it would be, catabolic would be, of course, hydrolysis. In which case would it be spontaneous? Catabolic or anabolic? Spontaneous. This would be not so spontaneous. Cool. In which case are you making polymers? So make polymers. No questions. Break polymers. Yeah.
What's it? Catabolic reaction, how anabolic reaction, just like this reaction.
Coupling. Go Mustangs. Yeah, good. Oh, so I guess I could do like a little drawing like this. We're gonna say what happens if you mix the two together. And then I guess you could say that would be coupling reactions. That works out really cute. I like that. Right? Like for instance, me in chill state to get me in run state. Is that going to be a spontaneous or not spontaneous process? Appreciate y'all very much. So obviously, if I need to make this happen, I'm not saying I need to, let's just pretend that I wanted to make this happen, I would have to couple that with ATP hydrolysis to make ADP and phosphate groups. Yeah. So I'm going to break down my energy, right? That means that later on, after I go run my 12 steps, and then I decide that I've done a good job and I go hit up the CC's buffet, I'm going to have to remake my ATP, right? So I can go run maybe 13 steps, and I have to couple the synthesis of ATP with this concept of cell respiration. So taking glucose basically and breaking it down into carbon dioxide and water. Yeah, that makes sense. It's really, really cool stuff. Think about it. Good stuff. I like that. Thank you. Osmosis, which is I think a good topic as well. I think we want to remember some simple logic. So let's draw out our beakers again. Let's put our little cell in there. A lot of you guys have been signing your notes for, passing a class or whatever. So you've all seen my signature, which contains the solute particles. Awesome. In which case do we have an isotonic solution? Okay, so this would be an isotonic solution. Okay. In which case do we have a solution that is hypertonic? Okay, so this would be a hypertonic solution. Remember, there's just too much going on in the solution. So there's a lot of solute particles, which means that this would be a hypotonic solution, right? Now, if I was to reverse my wording, and I hope you're all paying attention here, I'm going to talk about the cell now. The cell actually would be hypertonic cell. This is going to be an isotonic cell. That never changes. And the cell here, of course, is going to be hypotonic. Do you see the direct reversal on these guys?
Usually whenever it's one question, they'll usually say
Most of the time. Like 9 times out of 10. Yeah. What is this? What are we moving in this case? Just to throw this in there.
Water.
water moves, right? So we want to make sure that, of course, osmosis, we remember, is the movement of water, not the movement of particle. Only water. Okay, so water, of course, would go to the inside here. It wouldn't really do much there, and then I think it would go to the outside here. So you're right, and we're seeing that movement of water particles. Don't forget that there's other types of movement, correct? So since we're in that zone, let's talk about other types of movement. Like, for instance, let's talk about active transport. Is that going up or down the gradient? Okay, up gradient. Against the gradient, if you will. And of course, I would be using ATP, or energy. Let's talk about some other types of passive transport, like simple diffusion. And we'll also talk about facilitated diffusion.